we're going to break biology down into essential questions, and you are going to keep practicing getting better at answering these questions. And if you do that, it's going to improve your chances of being able to not only answer questions that rely on recall alone, but also it will give you a better chance of being able to apply the biology to different kinds of questions. So the first thing is, what's the point of microscopes? So remember that in biology there's something called the cell theory, okay? So it's this idea that living organisms are made of tissues, organs, and tissues are made of cells. Now, microscopes produce magnified images, okay? Microscopes produce magnified images that are not only bigger, but at a high enough resolution, okay? Remember, there's no point in magnifying something if it doesn't increase the detail with which we can see the structures, okay? So we're producing magnified images, we're producing them at a high enough resolution that gives us more information. And these things allow us to view the biological structure, okay? So because we can now see the biological structures of things, we can see, we can look at the structures of tissues, how the cells are organized. We might be able to even see what the shape and structure of the cells are, maybe some of the structures inside the cells. Because of all that stuff, we now have information with which to connect biological structure to biological function. Okay, that's the point of microscopes. It comes up quite a bit, but that's what they do. Our second question was, how do they do that? How do microscopes work? Okay, microscopes work. Generally, there's a number of aspects that we need to look at, but stage one, okay, so I'm going to break this down into stages. Stage one of how a microscope works is, first we need to prepare our sample. Okay, so samples need to be prepared. Preparation of sample, right? Different microscopes specialize in looking at certain types of samples. Once we've prepared our sample appropriately, then we can move into the actual producing the magnification of the images. So once we've prepared our sample, then what we do is we excite the sample. So we place the sample in the microscope, and in a light microscope we're shining light on it. In an electron microscope we're bombarding the sample with electrons, and so on. So we excite the sample with something, okay? And three, what we do next is when we excite the sample then, that some of that light is reflected, or some of those electrons are bouncing back up off the sample, or the electrons are going through, which results in emissions. And the magnification happens when we change the direction of those electrons. So we produce some emissions. So whatever's bouncing back off the sample or going through the sample, those emissions are refracted. We're changing the direction of them through lenses in the case of light, magnets in the case of electrons. Those emissions are refracted to produce the magnification, okay? Now, the excitation affects the resolution, okay? It depends on the wavelength. That's going a bit too much into the detail, but the excitation will affect the resolution, and when we compare the microscopes, this stuff will be important. The emissions refracted, the extent to which we can bend these emissions affects the magnification we can achieve. So that's us talking about magnification. Okay? And four, finally, when we detect these refracted emissions, so in a light microscope, basically those bent light rays or light waves are entering our eyes, and we can see the image. In an electron microscope, those electrons are detected, and that helps us to produce an image on a screen. Those emissions are detected, and when those emissions are detected, it affects the type of image produced. This is how a microscope works in general, and we can use these things to compare the microscopes, their advantages and disadvantages, etc. Okay, that takes us to our last question. How can microscopes be? And what we're going to do is make a table. Okay, didn't use a ruler, that's naughty. And what we're going to do is we're going to use the categories that we spoke about before. Sample prep. So we had sample preparation, we had the excitation, okay? We had the refraction, so we had the process of magnification, I'll just call it that. Okay, and we'll also talk about resolution here. Magnification, resolution, so we'll make a comment on that, yeah? And essentially with a view to comparing these. So it's not just about knowing that optical or light microscopes are excited with light, visible light, it's about how it compares to the other ones, yeah? And so we had sample preparation, excitation, magnification, resolution, and finally we had detection, which directly connects to the types of images produced, right? And we want to look at each of these things as a positive or a negative, advantage, disadvantage, etc. Okay, so let's get into it. Sample preparation optical microscope is easy. Okay, we are going to get into light microscopy in a bit more detail later on in another session, but for now we'll just say it's easy, it is cheap, okay? You can use stains, so there's real colors. It shows you the real colors of objects. Does that go in images produced? Yeah, that goes a bit more in images produced. So I'll take that off. So sample preparation is easy and cheap. Images produced, real colors. Okay, the samples here can be living or they can be dead, no problem. And sometimes we can use stains, right? Differential stains, we can use those to enhance the images, okay? And those would be false colored, so that wouldn't be real colors. Okay, I think that's fine. And excitation, we're using visible. And then the magnification and resolution is, I think the magnification is around 2,000 times, and the resolution is around 200 nanometers, which is relatively low, right? So it's comparatively, relatively low. Okay, images produced, they can show real colors. I guess you can say that they're magnified, but they're not as highly magnified as what you might get with electron microscopes. Images produced can show living samples. Yeah, so you can actually see things happening because you're looking at a real living thing, right? So there's less artifacts. Okay, let's quickly move on to laser scanning confocal. Sample preparation is not so easy. So you can, it's not as easy. So sample preparation, so these samples can also be living. You can use stains, but usually they're called labels. So you can label very specific things in the cell. Yeah, it's not, I wouldn't say it's super cheap, but it's not as expensive as electron microscopy. So So cost is kind of medium. And in terms of sample preparation, it is not as easy as light microscopy, but it is not as complex and difficult as electron microscopy either. Okay, in terms of excitation, the laser scanning confocal microscopes uses focused visible light. So essentially that's lasers. It's still a type of visible light, but instead of like the light rays or the photons going in various different directions and being of different wavelengths, here we're kind of focused in terms of the directionality. So, okay, so that's a laser as opposed to visible light. Okay, so it's still visible light, but it's focused, and the direction of all the beams of light is the same. Right, now in terms of magnification, theoretically, the magnification is still the same, and the resolution, in theory, is still limited by the fact that we are using visible light. So theoretically, the resolution should still be around 200, but it is an improvement upon the optical or light microscope, or basic light microscope. So maybe the resolution, we could say, is slightly less than 200 nanometers. So if that's 200 nanometers, this is going to be less than 200 nanometers, i.e. it's a higher resolution than the light microscope. You don't get real colors, so no real colors, because everything that you see here is from the labels that you have added. Okay, so no real colors. So we see labels, so colored labels are added. Okay, so you add these labels, and the labels attach to specific things in the cell, and then when you shine the laser on them, those specific things that you have labeled, they can be viewed. Okay, but yes, you can see living cells. Okay, you can see detailed structures within the cell. So structures which are not visible here can be made visible here. So I guess we can put here ultra. You can see some ultra structure aspects. So a cytoskeleton, for example, specific proteins, even in the cell, specific organelles. You can see those things, whereas you might not be able to see them in light microscopy. Okay, electron microscope. So sample preparation, we're going to put these together. So in terms of electron microscopy, the sample preparation is complex. Okay, it is expensive. It requires technical expertise. Right, whereas especially light microscopy is very, very accessible. Everyone can do it, pretty much. Confocal microscopy needs a little bit more skilled preparation, and electron microscopy still further. Okay. Samples have to be dead because they're produced. The samples have to be viewed with electrons in a vacuum, and in a vacuum you can't have a living sample, to my knowledge. Okay. Right, and that then takes us to excitation. Again, this is similar. Actually, we can separate it. They are both electrons. They are both excited with electrons, but in terms of scanning, the electrons bounce. We're talking about electrons which are reflected off the surface. So that affects the kind of image that we're going to get. Whereas in transmission electron microscopy, we're looking at electrons transmitted. So these electrons, we're looking at the electrons that make it through the sample. okay, which again, it affects the type of image that we're going to get. In terms of magnification, I think these are both really high, so these are much higher magnification. In terms of electron microscopy, we're talking about magnifications in the range of millions. Okay, so I've seen 2 million quoted, sometimes even higher than that. Though that magnification can be as low as in the range of 50, 100, thousands, but it can go up to times millions. Okay. In terms of resolution, the resolution is very high, 0.5 nanometers, okay? So that is much smaller than the resolution for light microscopy, therefore it's a higher resolution, okay? So we're able to see with much more detail, okay? We're able to distinguish much closer points. Okay, in terms of images produced, the images that are produced with electrons are black and white, okay? That means they can be false colored, so you can produce colored images, but they're not showing real colors. Those colors have been added post image production, okay? So false color images, they're black and white, they are very detailed, okay? So you can definitely see all the ultrastructural components, right? So you can see ribosomes, you can see proteins in membranes. I mean, you can even see the phospholipid bilayers, right? You can see the cytoskeleton components. You can see a lot of detail in the cell. However, your samples are always dead, and there are possibilities that because the sample preparation is so complex, you might affect the sample before you view it, i.e. creating kind of possibly things that are not real. Okay, so artifacts are possible. Okay, so there we have it. We've made a comparison of microscopes, and hopefully it didn't take too long. Okay, it's rough and ready a little bit, but done is better than perfect. So that's our 3 questions. Let's just quickly recap what those questions are, and you can have a chance at doing this for yourself. Number 1, I think I phrased it as what's the point of microscopes, but what do microscopes do? How do they contribute to biology? What's their importance? There's many ways we can word it, and the examiners will be playing around with how they can get the same information by asking you the question in various different ways and disguises. But what do microscopes do? How do they contribute to biology? Can you answer that question? 2, how do microscopes work? Now, this is a question that I've never seen it come up in an exam, but this question forms the basis of so much other stuff that you have to know. Like all that comparison that we did with the microscopes, ultimately, the way we organized that table was by understanding the key components of how microscopes work. These are your 3 questions. This is now where you get to work. And now I get to work and see if I can edit this video down to 15 minutes or 10 minutes ideally, okay? But the point of this, remember, the point of this is not, if this is your head, okay? Right? The point of this is not the information going in. That part has already been done, and you probably just need to brush up on it a little bit. Where you might have been having more difficulty is being able to reproduce that information. So taking it from your brain, right? Taking it from your brain and being able to put it onto paper. Right? So if it's all this all the time, you can see why that's going to be a problem. Things only get stored in your brain if there's the input followed by the output, okay? The encoding and the retrieval, all right? And once it gets stored in your mind properly and solidly, then you'll be able to reproduce it in an exam, okay, and get that grade that you're aiming for, okay? And that's the transfer. All right, okay, so I get to work editing, you get to work doing that. Good luck.